The following is a conversation with Michelle Beckwith. Shelley is the co-founder of Paternity Testing Corporation and has been on the cutting edge of the human identity business for over 26 years. Shelley's cutting-edge technology has been used by numerous government agencies, including Health and Human Services, Department of Defense, and Customs. She has been instrumental in the victim identification world and has been involved in criminal paternity and child support services in over 5 states. The genetic testing that she's been able to develop has also been very helpful in identifying victims of natural disasters, such as Hurricane Katrina. Recent advances in genetic testing are also allowing for personalized medicine that will be affecting more and more people in the years to come. This is a Dr. Gil Show podcast. Now here is Michelle Beckwith. Welcome to the Dr. Gil Show. This is where we talk about medical matters that matter to you. My guest today is Michelle Beckwith. Welcome to the show, Michelle.
Thank you. It's great to be here. Michelle
is the founder and one of the scientists that started Paternity Testing Corporation back in 1996. Mm-hmm. Sounds about right. It has gone by the initials PTC for many years. So, Michelle, how did you get involved in genetic testing?
Well, so initially I started working down at WashU in St. Louis on the Human Genome Project. Of course, I was a chemist, and genetic testing was pretty new then, or at least PCR was pretty new then.
PCR, that's where you can amplify DNA and really start measuring it.
Right. And so that was the beginning of the Human Genome Project. And I kind of got sucked into genomics then, and I just never left.
And you never left. Now, your company has done just amazing things. I think we can call it, to use an overarching term, it is human. You're in the human identity business.
Yes.
Which is essentially forensic technology to identify human beings through DNA. Now, forensic means the scientific investigation of crimes. So you've been involved in all kinds of interesting things that I hope to explore as we chat today.
Okay, we can do that.
So let's talk about generally the type of stuff your company has done. I understand that you were the government, you had lots of contracts with government agencies to identify victims of crimes, family members, identity of people for child support,
mass disasters.
We involved in rape kits, all this kind of stuff where the genetic information is vitally important for the government, for companies. I understand you've even worked for insurance companies.
We have.
We have. So maybe you can find out who was driving a car or something after a crash, and all you have left are remains.
Or even sometimes it can be the result of trying to figure out who was the driver of, say, a drunk driving accident, or it doesn't necessarily have to be a fatality. It's trying to understand who was driving the car.
I see. So this is an incredibly important work that you do. Now, PTC has branched out over the years, and we're going to talk about these various things your company does. I understand you can test a person's genetics to see what medications might or may not be appropriate for them.
You can. It's actually called pharmacogenomics.
Uh-huh.
And so what we actually do is look at the genetics that helps generate your liver enzymes.
And your liver metabolizes medicine sometimes quickly, sometimes slowly.
Mm-hmm. And we can, by looking at the genomics of your body, we can actually determine what your liver enzymes will look like. And if we know what your liver enzymes look like, we can do a pretty good job of predicting how you're going to metabolize a lot of them, not all of them, but a significant number.
And it can offer personalized medicine for someone so that a doctor can choose maybe a dose or one drug versus another.
Yes.
Fascinating. So let's go back to the beginning, Michelle. You were involved in the Human Genome Project, which for our younger viewers has probably been a part of life the whole time. But when I was growing up, mapping the human genome, meaning determining the sequences of DNA in an average human being, was very important. We didn't know. And the older technologies were slow and cumbersome, weren't they?
They were.
Really slow, and they were estimating it would take so many years to do it. And then some troublemakers came in like Craig Ventnor and whatnot.
He did.
He came in and said, whoa, we can blow this apart. We can shotgun this, do massive parallel sequencing. And I think
we can patent everything along the way.
And patent it, yeah. And make a lot of money in the process. But a brilliant guy. Maybe not the most politically suave person, let's say.
Yep.
And my audience needs to realize that they were all a little different too. So they needed the basic pattern of the DNA, realizing there's lots of spots where we differ, which makes us unique. And I think they tested 5 people in the beginning. They wanted to get 5, and Craig Ventnor was one of them. He put his DNA in the pot to be analyzed. So you got involved in the Human Genome Project. And then what gave you the idea, said, hey, I can do this for a living. I can make a business doing this. Tell me how it started at PTC.
So I really wanted to work in forensics from a very young age. But I always thought I'd end up in chemistry. So my little trip into WashU changed all that for me.
Well, when did you get the idea that this would be so valuable for so many people?
Well, so at that point in time in history, that was just the beginning of DNA in forensics. We were still using HLA and ABO blood grouping for paternity testing.
Okay, these are proteins on the surfaces of cells, and with antibodies and various colors and enzymes hooked up to it, you could get some basic idea of a blood type or the genetic makeup of someone to help at least say, hey, this is more likely a suspect, but it wasn't 100%.
Right, and at that time, we really couldn't use, on the rare case, you could use ABO
blood type. Okay.
blood typing for a forensic case, but it was predominantly the only tools available for paternity testing. And so there was this huge need. We were starting to transition small tools, small pieces, one locus at a time, so one DNA location at a time, into the forensic world during that time frame. But there was a huge need in the paternity world.
Right.
And as people were starting to transition into genomic style testing for paternity, there was just a huge hole in the market. And my mother, who's also involved in PTC, had already been in a crime laboratory for several years and was very involved in a lot of the validation of the new DNA testing.
So you founded this company with your mother?
I did.
Wow. I assume you two got along for the most part.
Yes.
That can be difficult. So when there used to be a show run by a host named Maury Povich, and he was famous for opening the envelope and saying, you are the father, or you are not the father, and pandemonium would break out on the set, and fisticuffs and all that kind of stuff. Was that did that come about because of the DNA testing? It was so accurate, or were they doing paternity from these blood group types and stuff back then? Do you recall?
No, I don't think that there was any of the paternity testing showing up on any of those type of TV shows before we could do genetic testing.
And really be sure.
Yeah. Not only be sure, but, turnaround time mattered, getting cooperation. The sample types mattered. So,
like blood versus saliva or skin or something like that.
Right. So cheek swabs really changed who was being able to be, or willing, not able. Willing to be tested. The number of people, and I'm sure you know this in your world, that the number of people who don't want to have a blood draw done is pretty high.
Yeah, and it's usually the guys. The women take it. The guys are the ones we got to lie them down because they're going to pass out.
That's right.
And all this stuff. Yeah, so you can get this DNA now from a cheek swab and whatnot. And you're talking about turnover time. Like how long would it take in the early days to get a turnover? A couple weeks would you take to get a result?
So ABO blood grouping wasn't quite as slow, but we started something called RFLP testing, which is a long word I'm happy to describe.
Right.
But really what it is it's a very accurate method of DNA testing, but it would take a week and a half to 2 weeks to get results back. It was a tremendous amount of laboratory work. Over time, we moved to some systems where we had to use a lot more because they weren't each individual one wasn't nearly as reliable, but it was much faster. We could get results back in 24 hours if we needed to. And that's when we started to see a lot of the TV shows start to join in. Because something that takes 2 weeks, a lot happens in 2 weeks.
Sure. Person runs away and whatnot. And I'll just make a comment here. You watch all these cop shows nowadays, and the first scene they're at the crime scene, and then they have the guy in the interrogation room like 2 hours later and said, "Your DNA was found at the scene." So clearly way too fast. It's not as dramatic, but
we're starting to get into that these days.
Really?
So about 10 years ago, there was DoD contracts and DHS contracts.
Department of Defense and Department of Homeland Security?
Awarded to several contractors, and the goal was called rapid DNA. So rapid DNA, so ultimately we didn't meet the requirements as a community. We didn't get to meet those requirements, but we came close. And so what came out of all of those contracts was basically a laboratory in a box. So we have a box. The idea was to try to make this swab in, profile out, searchable CODIS profile out. Now, CODIS is the database. It's called a convicted offender database. So the idea was that, say, in a booking station or at a border crossing or in the middle of the desert, we could actually put this box in that anybody could use. We could have a profile in, we were hopeful for 45 minutes. It's more like an hour and a half. But ideally, in good circumstances, it can produce a DNA profile that is, in some cases, searchable against our databases. And so the idea is to try to not let someone leave a booking station if they've found, a serial rapist. Before they leave. Or to be able to connect families at the borders when they're coming in. So if somebody's bringing a child in, they can confirm that child belongs to the mother.
And not being trafficked.
Right.
So you have this the goal was a portable lab, a box.
That could be used by anybody.
Anyone could use.
DNA typing's complicated. It's not perfect, but it's really good. And today it's getting better.
I know when you get paternity testing, it's never 100%. It's like 99.2 or 98. And I think the term in legal is it's practically proved or something like that. Good enough. If it's 99%, you're the father, right? You're the mother.
I really like to see 99.99%.
All right.
But for practical purposes, it's the difference. So that statistic is a little bit misleading. Because we think, and kind of the same way in your practice, like 1 in 100 happens, right?
Sure.
And a 99% is 1 in 100 people, approximately 1 in 100 people would match, and you do. Does not include any of your relatives. So there's some complications there as well. You end up with and today technology is good enough that there's no reason that we should be settling for 1 in 100.
You can do better.
We can do much better.
Interesting.
And also in the process of doing better for, just the individual, so a 99.99% is 1 in 10,000. A lot more confidence between that and 1 in 100. And they sound like the same number, even though they're not. When you talk about them in terms of percentage, they're not even close.
Many more decimal points over. Right.
But it also gives you a little bit better reliability, usually into relatedness as well.
Gotcha.
So potential uncles, potential grandparents, things like that.
Yeah. So when you, going back to the beginning, when you were developing these paternity tests, what types of agencies, or you already mentioned DOD and Homeland Security, but earlier on, what type of people would contract with you for these paternity tests, and what kind of cases were they looking at? Give me some examples.
So child support enforcement is a big contractor of paternity testing.
Yeah, 'cause that's money. That's obligations.
Right.
Right.
And it also helps shift families off of government resources into often better situations, things like health insurance, things like that they may not be getting sufficient support from the government, and they can transition to a parental support, which is good.
So you can put some weight on somebody, say, you are the father, you are the whomever, you need to help support this child, usually, I guess.
Well, and it also helps bring families together. I mean, you'd be surprised how often people don't know.
Oh, I didn't even think of that. It's like, oh my God, I didn't realize I had a child, and
Right.
maybe that'll uncover some paternal instincts, some good things.
Yeah, a lot of good things.
Oh, that's great.
Then there's also, we've done a lot of forensic work as well. So we have contracts with police departments and with crime laboratories, both for chemicals to do their testing, as well as actually doing some of that testing for them.
So you would support crime labs as well with what are called reagents or the chemicals. Interesting. So they could have their lab somewhere, maybe a small town or something, where they don't have a university, and you could supply them.
Most states actually have a state laboratory, and some of them actually have multiple laboratories. So here in Missouri, we have Missouri Highway Patrol has a large crime lab in Jefferson City. They also have one in Springfield.
Okay.
And there's another one down in Cape
Girardeau.
And then some of our cities also have their own crime laboratories. Kansas City has its own crime laboratory, as does St. Charles, St. Louis County, and City.
Okay.
All have their own laboratories as well.
So your company, PTC, was helpful in supporting and keeping those labs running?
Some of those laboratories use one of our chemical products. We've always helped support with complex kinship statistics. So these are cases where people are related instead of direct matches, which is what a lot of crime labs do. They match person to person, person to stain, person to a mixture, but they don't normally do complex relationships. So do I have grandparents? Can I make an ID from somebody's two siblings? Things like that. They don't do the population statistics associated with that, so that we can put a number with how likely is it that this person actually is who we think they are.
So you don't have, I know, if somebody dies in a plane crash, I know they go look for a brush or a toothbrush that has some of their cells on it, so you could maybe match them. But if you don't have that, you can look at their kin and then get a statistical likelihood. Am I saying that correctly?
You are. And so one of the things that we started with the Missouri State Highway Patrol and Kansas in 2001 as a group, we started the Mid-America Forensic DNA Conference. And so most of the DNA analysts in Missouri, Iowa, Kansas come here every year to all work together. It's really bridged a lot of communication gap between a lot of the DNA analysts for everybody to be here together and talk every year. And be able to pick up the phone and, if I need help with a case, I know probably who to call in the 3-state region. If they need help with a case, they know who to call. And so that's been a really wonderful game changer, I feel like, for the forensic community in Mid-Missouri and the whole mid part of the country.
Well, that's fantastic. So you can establish networks of experts and I assume best practices, what's working best, and you can all help each other.
Yeah, that was actually really neat during Hurricane Katrina.
Tell me about that. You were involved in identifying like bodies or body parts or unidentifiable remains. Is that correct?
Yes, and during Hurricane Katrina, in most cases, it was unidentifiable remains. There was not what we call dysarticulation. There was not very much of that during Hurricane Katrina, a small amount, but not like you see like during 9/11 or some of the other disasters that
where
happened.
you're seeing body parts. There's just a little fragment of a person. So in Katrina, was that the body was decomposing in the heat and you couldn't tell much more?
Right.
And so what did you do for what agencies to help identify bodies in Katrina? What was that like?
So Louisiana State Police and then also the New Orleans Coroner's Office were ultimately tasked with victim identification. Of course, FEMA provided a tremendous amount of support, as did AFDL. And AFDL is the Armed Forces DNA Laboratory.
Ah, so you all worked together.
DMOR also came in.
What's DMOR?
So that's the disasters, a division of FEMA. It's a disaster mortuary services.
Ah.
So they're often the ones who provide anthropologists and dental guys who come in and help, because those are easier modalities to identify people with. I mean, there's less cost, there's less, they're faster. You know,
if you have a dental X-ray, you hold it up. Yep, there they are.
But so one of the things that happened during Hurricane Katrina was that all of the personal items, so you were talking about toothbrushes and things like that, they were all destroyed. Family members were scattered all over the country.
Oh, yes. I remember people in cars with all their belongings and the truck stops around. We would see them on the highways all around the country dispersing.
Well, and people had cell phones, but not everybody had a cell phone then.
Yeah, that was not everybody had it. Certainly didn't have a smartphone with GPS.
No, I actually think I had a Palm Pilot that I got on my way down there. Like one of the first generation Palm Pilots.
So you went down to New Orleans?
Yes, I spent 6 months down there.
Wow. Whoa. So tell me more of what type of work you were doing and what it was like.
So ultimately, all of the lessons learned from 9/11 were implemented during Hurricane Katrina. And so there were some things that went fantastic. It was a little slow, but we got there. And once everything was kind of online, we had all the testing was sent out.
Sent out.
So the actual genotyping portion of it. So the actual DNA test to get us a profile. We didn't have those cute little lab in a box things yet.
Right.
Was sent out to multiple laboratories to try to help deal with the volume.
Okay.
And so we were the central repository for all the data. So as all the laboratories were profiling samples, sending us all the profiles back. And then we were trying to reconstruct families. So we were collecting as many people, drawing pedigrees. So the pedigree is the information. It's a written description of what a family looks like.
Mm-hmm. And for people who don't know, it looks like a tree. Mm-hmm. Women are circles, men are squares, and there's these little lines. You got the parents, and the kids come down, and it goes up to the grandparents and the great-parents, and out to the uncles and the cousins and all this stuff. And you end up, there's this big tree called a pedigree. And I know there's all these mathematical formulas and probabilities that you use. So you construct a pedigree frequently.
And then we would decide who we wanted to collect. Who was going to be the best people available from this family to help us identify their missing loved one.
Okay.
So that was ultimately the goal.
Right, because you don't want somebody who's related by marriage. That's not going to help you.
Right.
Yeah, Uncle Steve may not be biological Steve. He's in-law.
Well, and it really takes... we wound up using a lot of geneticists who are genetic counselors. I said that incorrectly.
Sure.
And they're really good at eliciting that type of information. You know, there's still some social non-paternity. There's always going to be. You know, there's going to be people who don't know that their dad's not their dad. There's going to be fathers who don't know their children aren't their children. There are going to be grandparents who don't know they're not grandparents. It's just an issue.
We sometimes discover that in the fertility world. And as an aside, when I was in junior high, we would test our blood. You'd prick your blood thing and determine your blood type. And then you go home and ask your parents what their blood types were. And they stopped doing that because in almost every class you'd find out some kid, and it's not their daddy. And they'd find out by accident. So that they stopped doing that.
That was probably a good idea.
Probably a good idea.
But the genetic counselors are really good at trying to say, okay, can you sit down with me? Because if you sit down and say, who are your siblings? And they give you this list. But instead, they ask questions like, who is this person's mom? Who is this person's dad? Instead of saying, who are your siblings? They go through them one by one so that they can build a true biological pedigree. Because our social pedigrees often don't match our biological pedigrees.
Gotcha. Father-in-law, stepfather, right. Wow, so a lot was learned in 9/11 when we had just body parts in the rubble, and you carried that to Katrina. And what you've learned there, you can identify remains in other situations. Do you ever work with plane crashes or other disasters?
Yeah, we've worked actually all the way back to the RFLP days. We've worked plane crashes. So the RFLP was very, very early. It was some of the earliest genetic testing done in the late 90s.
Was that restriction fragment lengths?
Polymorphisms.
Okay, so you weren't looking at precise the steps in the ladder. You're looking at larger chunks.
Big fragments
of DNA.
Very discriminatory, though.
Yeah, properly done, you could get enough statistical power, couldn't you?
Very, very powerful. Each individual locus, each individual genetic location worth a tremendous amount.
And then you get the combination and you get your power from that. So you've worked with plane crashes, accidents. Now, who else wants this information? Do insurance companies sometimes want this information?
They do, and it can be for a lot of different reasons.
Well, tell me about why would an insurance company want to use PTC Labs?
So the normal reason is to figure out who's driving a car during a car accident.
That happens enough.
It does.
That's a thing.
It does. That's the average reason. But sometimes it is an identity issue, especially if there's no criminal investigation. So there becomes that inherent need to make sure that an ID is correct if there was no investigation done into that death.
And that does happen from time to time.
It
There's
does.
there's no obvious criminal activity, death of natural causes,
or a horrible car accident. But yeah,
or a horrible car accident. Wow. So, have there been any more advances in the field? You meant now. Do they have this lab in a box now, or it's almost a reality?
No, it's absolutely a reality.
Are there like Border Patrol people actually using it, or state cops using it? Or?
We do have some in some police stations around the country today.
Wow.
DHS has been working on trying to implement them at the borders for a long time, and there's been a lot of projects around that. I don't know what the current status of those projects is right now.
Now, do you know, I know a lot of kids got separated from their families crossing the border or anything. Are you at liberty to say if you've been able to help kids get connected with their parents?
Privately? Yes, not through government contracting. It doesn't mean that, but we have had people come to us who have been separated trying to put people back together.
Because I know if a kid is separated and they're little and 5 years later, they don't, you don't know how little Johnny has grown up, what he's grown up to look like necessarily.
Right. And we do see, the other thing that we do see is a lot of the homes and the shelters for children trying to place them with relatives of the United States.
And you can prove kinship.
Right.
That there, in fact, are a relative, can't you?
Right. And so we also see the same thing for immigration, not just at the border, but immigration from anywhere in the whole world, where, someone wants to bring over their parents. They've been here for 15 years and they would like their parents to join them.
Okay.
And so often what happens is they'll have to go through a process with the embassy to have their parents collected in their home country, prove that they're their parents. and then they can initiate the process of bringing them here.
So you can help in that situation as well. Say, yeah, these are my parents coming over, because they may not have picture IDs and the same type of documentation we have here, obviously.
And even if they do, it's often not accepted.
But they'll accept your testing sometimes.
Right. And so there's a lot of regulation around that. It has to be done in an accredited laboratory. There's not very many of them, and it has to be done directly, so there's nobody in the middle. There can't be any like resellers or anything like that, because immigration's trying really hard to rein in all the things that can go wrong along the way.
Yeah, because I'm sure somebody could slip in a sample. I mean, yeah, so you have to have chain of custody and witnessed, and I'm sure the whole thing
and now
needs to be verified.
Samples in other countries now have to be collected at the embassy by embassy staff usually. Or they can be collected by a contractor on the embassy presence, with people from the embassy witnessing
certified,
that.
protocols,
right,
bonded, all that stuff. So, wow, that's fascinating. Now, are there any new things in the technology that are making it more accurate or faster or cheaper? I know they go from, we talked about the RFLP to actual
STRs is what we use today.
What is an STR, please?
It's a short tandem repeat. So RFLP was a very large repeat. So think 100 base pairs or more repeated over and over and over again.
Okay.
A short tandem repeat is usually only 4 or 5 base pairs repeated over and over again.
And there's enough of those and enough variation to make identifications.
Right. And so to kind of give you an idea of the difference in power, so in RFLP days, we would use maybe 4 DNA locations. Today, in STR, so because they're much shorter and they're a lot more common in the population, they aren't nearly as powerful on an individual basis. Getting to 16 of them got us about equivalent power of the 4 older RFLPs.
Okay.
Today, we're doing 23.
23. So when you do the math and whatnot, all of a sudden that's much more powerful.
It is.
And it's quicker. So I thought, I'm not going to get into all the DNA mumbo jumbo in here because I want this to be of general interest, but that's enough to understand that it's very powerful. And that's how you can drill down to the DNA. Now, there's always this question of contamination. You find a body floating around in the floods of Katrina, it's got DNA from all kinds of sources on it.
Yep.
How do you know it's the DNA of the deceased individual?
So there's things you have to do to protect and safeguard against that.
Okay.
During Hurricane Katrina, what was collected was bone.
Bone. Teeth? You ever drill into teeth?
Well, I have, but not then.
Not then.
And it's certainly not my favorite, but it's doable and it works. But there are things you can do to clean up samples, in particular in a situation like that. But ultimately, in a water situation, water and bacteria destroy everything. Right.
Ashes to ashes, dust to dust.
And so typically remains that have been recovered from water, often, it's depending on how long they've been there, but are often, bone is the only sample we can test reliably.
Only place DNA has survived. So those are some challenges, and you have ways to get around that.
Well, bone sample is easy to clean as far as because you can sterilize the outside of it and take from the inside. So that's helpful. When you're talking about other body sample types, there's usually such an overwhelming amount of the actual individual that you're looking for. Contamination is not an issue.
Gotcha.
Because at about 10 to 1, 15 to 1, you can no longer see. So if you have a mixture of 2 samples,
okay,
where one is 10 times greater than the other,
okay,
you often don't see the other. You won't see the one that's only the one.
But the minority
right.
gets overwhelmed.
Right.
And doesn't contaminate the whole thing.
But so where you do have problems with contamination is often in forensic samples.
Right. And that's the segue I wanted, to go into the rape testing. I remember you and I have been talking, and we actually years ago collaborated a little bit on possibly testing some new technologies to help pull this DNA apart.
And you guys were tremendously helpful in doing that.
I appreciate that. It's nice to be able to give back. So now we're going to talk about something maybe a little gory. If a woman is raped, there are body fluids from the woman, there are cells from the woman. Let's say she was raped by a man, and there's body fluids involved there. So you've got cells from two people, maybe more, contaminants and whatnot. And when one does a rape kit, I was an OBGYN resident, and one of the jobs was to come down to the emergency room and help examine and evaluate these, usually a young woman. And you take a Q-tip in the genital area and you take a sample. Now that Q-tip is collecting cells, like I said, this whole mishmash of sources of DNA. And I believe you have been instrumental in developing a technology for separating that. And I think you call it erase sperm isolation.
We do.
Tell me, where did that start? Where did you come up with this idea? How did you develop this technology? Give me the whole backstory, please.
So one of the things we call differential extraction. And what differential extraction means is basically this process of separating cells and trying to get one cell type versus another.
Because we want the cells from the perpetrator,
right?
Because we want to be able to identify this criminal potentially.
And the easiest way to do that is separate epithelial cells from sperm cells. Because
let me stop there. Epithelium is skin.
Right.
The vagina is called a mucosa. That's wrong. The vagina is actually an epithelium. It's a skin, just like your skin anywhere else. Here's a nice little fact. Did you know most of the dust in your house, when you're dusting your house, that's mainly dead skin cells that have gotten in the air and they float around and whatnot. So our epithelia sheds these skin cells, and a vagina is going to be shedding skin cells like any other, like a penis is going to be shedding cells. So you take this swab, you're getting these cells from both. So epithelial cells from the victim, from the woman, is not what you want.
Well, and we ultimately sacrifice the epithelial cells from the sperm donor as well, because we exploit, for lack of a better word, the fact that epithelial cells are actually very easy to break open.
And it's called lysis. So you can lyse or break open the skin cells, the epithelial cells that might be from the victim. And are you left with the sperm cells concentrated potentially?
That's the idea. Sperm cells are the actual shell of a sperm is much more difficult to break apart or lyse.
Yeah, they're tough. They're small and they're concentrated. They're meant to be a little DNA bomb, in fact, to be able to get through all kinds of hostile environments. That's how they've evolved to function. So you can erase or get rid of the skin cells and, in fact, find those rare sperm cells if they're there?
Well, and this technology had been around for a long time before erase was developed. So what we would do is lyse those cells with a very gentle detergent. We would centrifuge the sperm pellet to the bottom and literally by hand with a pipette, as an art, truly an art of the scientist, pull off that top layer. And then we would try to wash that sperm pellet. And wash away any remaining epithelial cells.
So let me describe this. These tubes have a little funnel at the bottom. They're little cone tubes, so you could put the sample in there.
I should have brought one.
There's little. They can be big, but they come down to a cone. So you take that swab, that Q-tip, mix it up in there, add some detergent. I assume it's proprietary technology. And then you put it in a centrifuge. You put it in something that goes around and around really fast, and all the dense cells like the sperm, are going to get pushed down to the bottom and concentrate in the bottom of this tube. And we call it a pellet. Yep. And we pellet sperm, we pellet all kinds of things and you're telling me a tech can suck the fluid off the top with a pipette and leave a little pellet with sperm in it.
And then we try to wash that pellet. And then we've been doing that for years. Many, 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 many years. And so that process of differential extraction, as you can imagine, is dependent on the analyst and how good they are at this art of getting down to the bottom.
It's technique dependent.
It's also very dependent on what that original ratio is. If there's only, imagine if you collect a vaginal swab that's covered in mucus, it's covered in all this stuff, and there's only 50 sperm cells in the whole sample.
And they are tiny. So you're talking thousands or millions of epithelial cells and just a few dozen, maybe, sperm cells.
Right. And that presents big challenges. It's impossible in this situation because you will never get 100% of the epithelial out this way. It's just impossible. You can get close.
So you're enriching the sample in the sperm cells.
Right. And so one of the problems, there's a detergent that's been around for years. SDS is what we have always used, differential extraction.
That's sodium dodecyl sulfate.
Yeah, it is.
To standard soap. That is standard soap.
We actually use a little bit milder soap, but the issue is an enzyme. So ideally, we kind of think about this. Wouldn't it be better if we could just destroy all the epithelial DNA, and then we don't have to worry about this whole cleaning it manually?
Because we want to get rid of the DNA, because we want the sperm DNA.
Right. We just want the sperm. But if we started at 10 million to 1 or more, we're never going to get this down to we're down to 10 to 1 unless we can actually get rid of that epithelial DNA. Because this stuff sticks to the sides of the tubes, it sticks to the sperm cells, it sticks to everything.
Sticks to everything, yeah.
And so you're never, by manually washing this, literally taking water to the top of it, washing the pellet. Every time you wash that pellet, you lose more sperm cells.
And you're losing sperm too.
So you're constantly fighting this ratio. But the issue, DNase. That's what DNase's job is in our world.
DNase chews up as an enzyme that chews DNA into little pieces.
Right. And all of the detergents that we use, and we have historically used and tried for differential extraction, kill DNase. And so we were ultimately able to find a detergent. There a group of scientists, this isn't me, but a group of scientists ultimately found a detergent that allowed for the DNase to survive.
It didn't denature the enzyme. So the enzyme could function, and you could lyse and chew up the epithelial cells. So you came up with a system that was reliable.
Right, and so what we ended up doing in my laboratory was taking the initial research and making it work much better. So we went through and found, the best environment that we could to protect the sperm cells, because that's one of the things that can happen with an enzyme like that. It can cross the membrane of that sperm cell and destroy the DNA inside of it. So we had to figure out how to protect that, keep that from happening. And then we were ultimately able to kind of find the best conditions where we could get down to cases that only had 20 and 30 sperm in them.
Oh my goodness.
And not just that, it totally changed the dynamics of how our sexual assaults were actually processed in the courtroom as well. Because of this issue, we couldn't ever totally get the female out. And yeah, there are really great analysts out there who are very, very good at this, but they're few and far between. And you get a great sample, yeah, that works too. But you never get a clean male profile out of one of these samples before this. So we always had a mixture. We always had to take into account that mixture and how other people could match that mixture. One of the things that erased changed in the forensic community was all of a sudden we were able to get clean, single source, one person male profiles. And so all of a sudden, not only was the process faster, there was not this analyst to analyst variation, but then all of a sudden, the statistical analysis of this case just got easier, a lot easier, not just a little bit, a lot easier. And then on the stand, it was a lot easier to defend. I know this profile matches this person.
So you could withstand the cross-examination and the dogged attacks from the defense attorneys doing their job. So you became powerful and reliable enough to be able to testify on the stand. Do you ever testify yourself as an expert?
Once in a blue moon.
Okay.
Not normally.
I've testified as an expert for some case, a murder case, and things that I've been. And I tell you what, it's a whole nother world being up there. It's quite scary. So, you drink your water and try to sound smart, you know. It's a whole world of the expert witness. So this erased sperm isolation kit is now used by a lot of departments. Is it standard of care now? Is it used all over the country or the world?
It is used all over the country. Crime laboratories still use a variety of techniques. I don't remember exactly where our last numbers were, but I think it was greater than 40% of the crime labs in the country.
Use your baby.
We're using erase or some variation of it.
I see. Did you ever get patents on that or anything, intellectual property?
Actually, 14 years later, the patent is issuing next week.
So that gives you 6 more years of use. Aren't they good for 20 years? Is that 21
like that.
years?
I don't know. It's complicated, but at
this
point, it doesn't really feel like that matters.
Right. I mean, that's the thing about drug development. You get a drug patent, but it takes 14 years to get it to market. And then, they only have so many years to make a profit. That's one of the arguments for how expensive medicines are. But that's amazing, Shelley, that you helped develop this technique that has helped solve crimes that otherwise might not have been solved.
Yeah, and there's been Dr. Chris Carson, in our laboratory is responsible for a tremendous amount of the development and the actual optimization of the race.
But you've been part of it from the very beginning.
Oh, yeah.
That's fantastic. Is there anything else about that technology? Has it spun off or does it use for any other crime situations, or this technology mainly just to get the sperm cells from everything else?
It is. It has been the same ideology has been investigated for other things. But ultimately, an epithelial cell is an epithelial cell is an epithelial cell. And, separating epithelial cells from each other is just challenging.
Gotcha. So your company also started doing this testing, this pharmacologic testing for medical management reports. When did you start getting into this personalized medicine testing?
So 2010-ish area is about when genomics started having clinical utility.
Okay.
So we were starting to use more genetic tests in the doctor's office for varying things. We were also starting to learn a lot more, everything from oncology all the way into medications and everything in between.
Sure.
And so in 2013, we started looking at pharmacogenetic testing. It was a way to have a really positive impact on people. And in some cases, change their lives completely, especially in mental health.
Because you could help them choose a better drug.
And so we were looking, as clinicals started coming, we were looking for ways that we could contribute and make a difference. And that seemed to be a really big one. I mean, we were doing some orphan disease screening, things like that. And yeah, that's important. Do not get me wrong. Right.
Orphan diseases are these rare conditions, and there's so few people that there really isn't the money or the push to study them and put all the money into when you could study something that's much more common. I see. So you started to get the idea that you can test the genetics or what's called the genotype of a person, find out how their body might metabolize a drug differently from somewhere else. And did you then figure, well, we know this drug is metabolized via this cytochrome, and we know this? Did you have was there enough legwork that had already been done that you could have leads and you could kind of guide where you went?
So today we're in much better shape than we were in 2013, as you can imagine. But in 2013, a lot of that research was done, but it hadn't been compiled in a useful way. I mean, there were hundreds and thousands of papers on varying drugs, varying genotypes, varying enzymes. And so for the most part, especially then, but even mostly today, what we look at is the liver enzymes. So we kind of look at that drug in, drug out, which is what your liver is predominantly responsible for. Can we understand how the liver processes that medication? And do we know anything about the liver enzyme from a genetic perspective? And can we predict what that liver enzyme looks like? And if we can, then we can maybe, if enough research has been done, try to figure out, or at least predict, how that person's going to metabolize that medication, at least from a metabolism standpoint. Now, we've also started to look at some of the transporter proteins, some of the receptor sites. Even today, some of the research behind those things is incomplete.
Oh, it seems dreadfully complicated, and you're going to have to slowly, iteratively build on each other's work.
And so in 2013 was the beginning of a group called CPIC, which is the Consortium for Pharmacogenetic Testing.
And does that involve universities or drug companies or all of the above?
Predominantly universities, where researchers were assigned individually as a group, but researcher groups were assigned drug gene pairs. So one drug or a family of drugs that are all related, and whatever gene is relevant or genes are relevant to those drugs. And that's what they studied. And they were able to develop guidelines and standards around how we would recommend dosing and how we predict those. And so over the last 10 years, we've went from, 2 standards to dozens.
Dozens. So if we have an individual with a certain condition, would a doctor, for example, just help me give me a case example where this could actually be used? Let's say you use a drug and would you say it's not working as well for this patient? Would you then do this testing, or would you do it beforehand? Is it to a point now, before you even give a drug to somebody, you might do this testing?
Ideally, I think it should be done preventatively. However, I'm not sure all of our insurance companies agree with me. So
Sure. We have we're in a constant war with insurance companies.
But yeah, it's very helpful. I don't know if you remember, but I had a surgery done, and I know that codeine doesn't convert for me. I don't make the liver enzyme that converts that. And I was able to communicate that to you and say, just give me Advil because, you can send me home with whatever variety of codeine you want, but I'm not going to take it because it's not going to work.
Now, does that mean your body chews it up real quickly, or it doesn't, and now you've got toxic levels? Well, what happens in you, if you don't mind sharing?
Well, for codeine is a prodrug. And so when you ingest it, it's not active. Your body is actually responsible for converting that to the morphine-like substance.
Right.
And so ultimately, if your body doesn't convert it, it will eliminate it other ways, through other pathways. And in that particular case, it doesn't normally create a toxic situation. There are drugs where that does happen, though.
Right.
So for me, it's just that matter of it's never going to convert. I'm never going to get the benefit.
So it doesn't work.
Right.
It just doesn't work.
Right.
Gotcha.
But there's other drugs out there. Demerol is one that comes to mind, which we don't use very much anymore, or maybe at all.
It was taken off formulary. I think it's back in limited uses now. Demerol is like morphine, but it's like a half of morphine. So it's technically not an opiate, but it's an opiate.
Right, but if it doesn't, it's one of those things where if it doesn't convert, it's toxic on the front end too.
Yes, and I think that's part of the problem with it.
Right. And so if we had that person's genotype, you actually could avoid giving it to them in the first place. And some of our NSAIDs, some of our better NSAIDs that have been taken off the market over the years have the same issue as well.
Or NSAIDs are non-steroidal anti-inflammatory drugs. I think Celebrex was that the one that was taken off the market, or which?
Vioxx got taken.
But Vioxx was one of these special fancy NSAIDs. Now, was it because some people didn't metabolize it normally? Was that the source of the toxicity, or the clots, or whatever the problems were?
Right, and so the enzyme that's responsible for most NSAID clearance. So you ingest, most NSAIDs are active when you take them. And so you're fighting that clock of how long does it take for your body to remove it from your body. And so when your liver enzyme that's responsible for eliminating that from your body doesn't work, or you don't have it, or for whatever reason isn't functioning properly, every dose of it that you take starts building up more and more and more. And so for people who were not clearing that, we call that clearance, but for people who were not clearing that medication from their body in an appropriate amount of time, and they were taking it continually, you end up with all the complications associated with high toxicity from an NSAID.
Gotcha. So this testing, are there panels now, or you just say, hey, get tested, and you get a whole panel there?
Yeah, and so we can look at some of the most common liver enzymes, and it manages to touch a pretty broad spectrum of our common use medications.
Gotcha. So you can get a big chunk of the most likely medicines you're going to use. Now, so who is using this test nowadays? So
there's several very different environments. So mental health, this is a really important tool for mental health. I mean, ideally, we wish we had better receptor site information and all this stuff. Like, is it going to work information?
I see.
But knowing if we're going to have drugs that don't clear and we end up in toxic situations and things like that is really important in mental health. And a lot of these medications go through some of these liver enzymes. That's a huge portion of the market who's using testing over the last 10 years. They were certainly early adopters. If you can think about mental health, they're, any tool, anything that's going to make life a little better, we're going to try it. Now, in our world, because what we've done was build a medication management platform. So we built tools to help take this really complex genomic information plus all of the normal drug pharmacy information and tried to combine it into one tool so that we can actually get all the drug interactions at once. We can get all the genetic information at once. So we're not, so our physicians aren't having to try to pull stuff from, I got to go look up a genetic interaction over here. I got to figure out,
it's a nightmare.
The 7 drugs they're on from 2 doctors over here. And I got to put this all together and see if it's still okay. And so when we started building GeneTrait, which is our medication management platform, the goal was to bring all of this to a physician in 30 seconds or less.
There you go.
Take an entire patient regimen, so all of their medications, incorporate lifestyle, incorporate drug interactions, incorporate genetic risk, incorporate lifestyle risk, all at once. And so you could plug another drug in, say, can I give this to my patient safely, and have all of this checked all at once.
Shelley, this is so powerful. So how often are physicians using this nowadays?
Because of the insurance complications over the last couple of years, that's been really hard for, your average primary care. Genetic testing is expensive. So if their patients have to pay about it out of pocket, often, I think mental illness. I think people who are disenfranchised, they don't have jobs or insurance or whatnot. And they can't.
And is Medicare, is the government health system going to pay for it? And it's a high bar to get them to pay for it because they're strapped,
Well, and Medicare actually has recently started covering testing much better. And by much better, I mean in more situations, for more diagnosis, for more drugs. And so in our situation, so GeneTrait in particular is really good in our long-term care because it's helping the pharmacists in the long-term care facilities, so skilled nursing, our nursing homes, not only identify duplicate therapies, so patients who are on 3 similar drugs or 3 similar acting drugs that maybe came from 3 different physicians.
Yeah.
But helping them identify that quickly, helping them identify the genetic interactions quickly, helping them identify, how do I describe this? Helping them identify multiple medications that go through the same enzymatic pathway that could actually be impacting each other.
Yeah, they gum up the works. There was a kinetic limit or whatnot. I mean, you can only process so much, and you could overload the system, clearly.
Right. So think about like Prozac, fluoxetine.
Okay.
it loves CYP2D6, which is one of our liver enzymes.
CYP2D6, I love it. That's yes.
It loves it.
A cytochrome P. All right.
It doesn't have to have it for it to actually be cleared from the body. So it's not dependent upon it.
But that's a preferred pathway.
But it's a preferred pathway. And if it's there, it is a hog. And so we call that inhibition. So at that point, a person who is on that medication, for all sakes and purposes, basically has no CYP2D6 for any other drugs.
I guess it's hogged. I see. I'm understanding.
So CYP2D6 is also that enzyme that's responsible for coding. So somebody who's on Prozac may not actually have codeine work for them. And so at that point, they may think for life that codeine doesn't work, not realizing that there's this enzymatic thing going on behind the scenes.
Gotcha.
And so we're trying to identify that as well. So these medications that are impacting each other through this enzymatic inhibition and induction.
Right, and induction, right? Because your body will make more. It kind of responds, doesn't it?
Well, and there are some pathways that you can actually make work better.
Sure. Interesting. So I think I guess what it comes down to is, can you convince the payors that a healthy patient is a cheaper patient and is going to outweigh? I'm thinking of the nickels and dimes here. It's going to be cheaper to do this test. We'll save you some money if we have a healthier patient.
Well, and if the role switches over every year, in the short term, it may not save a specific insurer and company, may not save a specific insurer money, but over the lifetime of that patient, or even over a 5-year time for that patient, the amount of money it saves is unbelievable. Mm-hmm. Even in just like things like hospitalizations. If you look at something like Plavix is cheap, and some of the other drugs are cheap now too, but they didn't used to be. And the insurers want you to prescribe Plavix. Because they don't
cheaper anticoagulant.
Right, but it doesn't work properly for 30% of the population.
Oh, it's that much.
And so how do we find out it doesn't work properly?
From side effects, complications, death.
Mm-hmm. Because Plavix not working, there's no really outward symptom of it not working. Plavix is a prodrug as well. It has to be converted to work properly. And there's no way to really, there actually are some protein tests you can do, but they're not being routinely done.
Right.
And so when the insurance companies or whoever is pushing, the cheaper drug, but we need to know, and I'm all for using the cheaper drug, quite frankly, but it's got to be used responsibly. And we have the tools to use it responsibly today. Actually, we have for the last 10 years.
Boy, that's powerful. That's really powerful. Are you finding this technology is being adopted more? Or it's got a lot of inertia. I'm sure it's really hard to break in and change prescribing habits and patterns.
It is, and you'll see in some health systems where there's been a big push, and you'll see in other health systems there's been a big push back.
Right. So, oh man.
Because often the reporting, so that's key to all personalized medicine. It's not just pharmacogenetics. But the reporting and the interaction and the tools that we need to be able to give somebody conclusive answers for something that's so complex that they're going to have to spend 2 hours going through books and whatever makes it no longer clinically useful.
Right.
We need those tools to be able to get everybody the information they need when they need it.
Yeah, and it has to be quick because I feel terrible for primary care doctors and physician extenders. They have to see 6 patients in an hour. That means 10 patients. Now, that's not 10 minutes with the patient. That's about 4 or 5 minutes with the patient if they can type quick and finish their note. So time, it might seem, you can look it up in 5 minutes, or you've got thousands of pages of data for you. That's not what you need. You need fast, accurate information to make good decisions.
Now, we've seen in the concierge practices.
Oh, that's a whole different world.
Well, yeah, they've been using this testing for years. And they continue to use this testing. And the better the tools get, the happier they are.
The happier they are. Because the happier the patients are, because they're actually paying on the barrelhead, like they say, a real fee for a real service.
Right.
And that's where you'll see the benefits of capitalism, because it drives performance and value.
And so we used to see, when pharmacogenetic testing in particular, but any genetic test has this problem. You get plopped 5 pages of data that you know nothing about.
Right.
So,
pages and pages of data.
And so I have to say laboratories in general have, at least, especially, not necessarily for pharmacogenetics, but for other types of genetic testing, have gotten better. And I hope you've seen this as well, where the answer's on the front page these days.
Right there. And and then there's pages and pages of all the negatives. Here's the 272 diseases they don't carry. Right.
So we've learned, but
it's getting better.
It's getting better. But these clinical decision support tools are critical to precision medicine actually getting better and deliverable by a normal primary care physician. Right.
Or a physician extender who, as we get older and there's fewer young people and fewer doctors, we're going to need more extenders and as much support as we can give them. So much the better, because I'm starting to wonder who's going to take care of me when I get older. We can talk about demographic changes and population collapse and all that stuff.
Yeah, we certainly can. There's definitely been a large shift also just in healthcare too, is where we're bringing people in, and who's providing that care today.
Fascinating. Now, is there anything else your company has done over the years? Any other technologies that would be interesting for our listeners?
I'm sure. But I can't think of anything right now.
Shelley, I know when we were talking about having this conversation, it was like you had so much stuff, and we were wondering, do we have enough time? And I'm trying to give an overview here, and at least hit the highlights that will be of interest to people.
Yeah, how we do on time?
We're doing really, really good. We still have a little more time. Now, a little bit about you. Your paternity testing corporation, I believe, is evolving, and you're going to be becoming Trait Technologies in the coming months?
We are.
Fascinating. And so I assume this technology is going to continue to advance. I'm sure more drugs will get on your lists, and more interactions will develop. And
we're actually working on that right now.
Tell me, without revealing trade secrets or proprietary information, that's a good final topic. Where is this going? Where do you envision this technology going in the next 5-10 years?
So some of it may depend on the advancements from our vendors. So these are the people who make our equipment for genetic testing. And what they're able to do, what they're able to provide. Because one of the big limitations for genetic testing, even today, everybody was talking about the $100 genome coming our way.
Right.
And we may get there someday, but we're not there yet. And all of the different technologies combined all have their strengths and weaknesses. So like a whole genome isn't truly a whole genome. And that's very much a misnomer. And so
explain that, please.
So we can't use whole genome testing for pharmacogenetics, because one of the things that happens, homologs. So what a homolog is 2 genes that are very similar to each other.
Mm-hmm.
And so when you do next-gen sequencing, and so how next-gen sequencing works is it takes small fragments and tries to put them up against a reference sequence to figure out what gene they are. But if you have 2 genes that are very similar, think about trying to do that.
So you don't know one is a pseudogene or one is active or not.
Right.
So you're swamped with information.
Right. And so it's called an alignment tool in the next-gen sequencers. They can't differentiate like what that piece belongs to. So that data just gets removed from your whole genome. Profile. So there's very specific things we have to do. We can do what's called targeted sequencing. We still do it on the next gen sequencers. But targeted sequencing lets us actually, before we go in and sequence, we'll go cut out the gene we want.
Is that something like exome? Like we want to know what's being expressed.
Yeah, exome is similar to whole genome, but it's the same idea.
All right, so you're going to go and you're going to take this little piece of DNA that you're interested in and ignore the
It's going to go on a
billions of
sequencer all by itself.
all by itself. Okay. And you can do that.
Yeah, and so then we know exactly what we have. But that technology is still really expensive, and the data analysis on that is still very challenging.
Yeah.
Now, other types of technology are letting us still do some of this in a kind of a high volume, where we are also getting very specific information. Because one of the problems with doing like whole genome sequencing, because I wanted to know what one gene was, I've got all this other information now. What am I supposed to do with it?
Right.
Am I responsible for analyzing that?
Right. Are you liable?
And what happens when, if I didn't analyze it, or if I did, now I may have an incidental finding. So if somebody asks me about one liver enzyme, and next thing you know, I'm telling them about a BRCA gene.
BRCA is a cancer, is a gene that it inhibits an inhibitor, and people can get cancers, higher rates of them.
Right. And so at that point, you have an incidental finding, and you're answering a question you weren't asked. And sometimes that's good, sometimes it's not.
Right. I didn't want, oh, gee, you have Huntington's. Oh, no, I didn't want to know.
Right.
And the cat's out of the bag.
And so there's this whole ethical issues that have had to be discussed in large community forums and things like that around, how do we deal with this overwhelming amount of data? Do we just only look at the things that we were investigating? Why we did the test? Are we ultimately responsible for keeping track of that large genome? And so there's other ways to investigate things on a much smaller scale. So we can do simple PCR testing. We've got fancier PCR testing these days that let us do a little bit more things.
PCR is amplifying DNA. So we got a little tiny bit, but if you double it and then double it and double it and double it, soon you're swimming in it.
That's right. But we can look at one specific place when we do that.
Okay.
And so we're not getting all of this extra information. So we're answering one very specific question. And so some of that technology, though, is each individual assay itself. So each individual DNA location we look at is pretty expensive to run. On an individual piece, they're not. But if you need 60 or 100 or say 2,000 or 100,000 of them.
Oh, boy.
Right. And it starts becoming cost limiting.
Sure.
To run that much. And so as we're getting newer technologies that are allowing us to get better data quality out of some of, and that's historically been some of the problems with things that give us lots of data, the data quality isn't that great. So that's always kept all of us in this little world of one assay at a time. But as some of the technologies that get us more data for less money improve, we're going to continue down this road of building tools that allow us to ingest this information and provide clinical decision support. It's going to continue. And
I see this is ripe for artificial intelligence. I bet you there's learning that can be done.
There's people trying.
Yeah, it's coming.
There's people trying.
It's coming. They're using AI in fertility, looking at how embryos grow and whatnot, and you just unleash it. You just let it look at whatever it wants to look at, and sometimes you see patterns and stuff's going to emerge from that. Wow.
Well, and it may help us continue to learn more. There's a couple of specific liver enzymes I'm really interested in because the research around them was kind of inherently flawed when it was originally done. And as a community, we've had a hard time fixing that. So my hope for the artificial intelligence is that we're able to rectify that situation.
Wow. So this is, it's an ever-growing fund of knowledge, ever-growing understanding in a field that clearly is virtually limitless and so complicated. I imagine people will be working on this for generations to come.
I imagine so. I can't wait to see the tools that they build.
It's going to be fascinating. Very exciting. Well, Shelley, I have learned so much. This has been such a fascinating conversation. It's wonderful to chat with an expert that has been been in some of this from the beginning. You've been involved in the basic science, the application, the ability to help solve crimes, and now you're helping treat patients, improve their lives, and help doctors and other healthcare professionals make very, very important decisions. So, Shelley, what a pleasure it's been talking to you. What a pleasure it's been working with you over the years. And it's a real pleasure to call you my friend. Thank you so much for coming on the show.
Well, thank you for inviting me. It's been a lot of fun, and it's always great to see you.
What a pleasure, my friend. Thank you so
Thanks.
much.